In last lecture, we have gone through the parameters like Pili, Fimri, that we got an information that Pimri and Pili, these are the associated for the attachment and for the movement. Sorry, not a movement, we can say attachment, and it is the useful parameter in a sexual reproduction or sexual related reproduction is carried out by this Pili and Fimri. So gram negative bacteria is having this particular type of fimri, and pileus are similar appendages about 1 to 10 per cell that differ from fimri in various ways. Say for example, they are genetically determined by sex factor, or conjugative plasmids are required for the bacterial mating, and some bacteria or bacterial viruses attach specifically to receptors on sex pili and at the start of their reproductive cycle. So basically we can say that pili and fimri are useful appendages or organelles for the attachment of the bacteria. So these are two most important things we discussed in last lecture. Now moving to the next parameter, that is the flagella and motility. Now as we look towards the living organism, and one of the most important category of the living organism is movement. So as we know that the bacteria is considered as a living organism, and that's why we can say that some of the bacteria shows the motility. They move from one place to another place. Now for the movement of the bacteria, the most important thing is required, that is the appendage or organelle for the movement. And second, the environment in which they or that particular bacteria will grow. Say for example, liquid or semi-solid, semi-liquid material is required for the movement. So it is not possible for the bacteria to move on the solid surface. So we can say that in a liquid or semi-solid surface, this bacteria will move from one place to another place. Now what they perform, or what is the organelle available for the bacteria for the movement? So it is nothing but the flagella, and that flagella helps them for the motility. Now obviously we can say that the flagella is having a different types. Say for example, sometimes bacteria will go forward, sometimes bacteria will move in a backward direction. So for these purposes or movement purposes, there will be a availability of flagella. So flagella is a motile bacteria moves by the use of this flagella. It is thread-like locomotory appendages extending outward from the plasma membrane and cell wall. So when we observe this particular bacteria in detail, we can say that it is the thread-like locomotory organ, and it will appear from the plasma membrane, and it is also appeared from the cell wall. So cell wall and cell membrane, these are the two parameters from where the flagella is originated. Now they are slender rigid structure, and it is measured about 20 nanometer across up to 15 to 20 meter long. So depending on the type of bacteria, we can say that the size and the length of that particular flagella is different, but the main function of this flagella is locomotion or motility. Now these flagella are made up of different parameters or different things. Say for example, flagellin is one of the most important chemical structure available in the flagella, and that is very rigid structure, that is slender structure and rigid structure, which helps these bacteria to move from one place to another place. Now depending on the type of bacteria or depending on the type of flagella available to the bacteria, there will be a different number of bacteria. Say for example, in some bacteria only single flagella is present, in some bacteria two flagella is present, or in some bacteria more than 2 or more than 5 flagella is also present. So depending on these, so here you can see in this particular diagram A, you can observe a single flagella is present, that is very thick type of flagella, and it is known as a monotrichous. So monotrichous flagella is present in some of the bacteria. Now in some bacteria you can see that both sides of the bacterial cell shows the presence of flagella. So for example, here on the both side you can observe the flagella structure is observed in very thin structure. You can see that flagella is present on the both side, so that is known as lophotrichous. So bacteria will move from one place to another place in by the use of this lophotrichous type of cell structure. Now in some bacteria you can see a single uh, flagella is present, but it is present on the both sides. So amphitrichous is the name given to this type of bacteria. And here you can see the last type is there, that is the flagella which is present on the all over the body. So here this is the image which is taken under the microscope. Here you can see the whole body of the bacterial cell is having the presence of this locomotory organ, that is flagella, and it will move in one direction by moving this particular flagella. So this is nothing but the structure, we called it as a flagella, which is present on all over the body of the bacterium, and it is helpful in the motion of the bacteria. Now when we see this particular type of structures in a bacteria, we call this flagella as a hair like structure. So it is very minute and it is observed only under the microscope. Particularly in a scanning electron microscope we can observe this hair like structure. Flagellal filaments are made of subunits of a single protein called flagellin. So as earlier we talk about this, flagellin is the flagellar filament. Flagellin is the protein which is present in the flagella, and that is a rigid structure. The transmission electron microscope studies have shown that flagellum is composed of three parts. So as we talk about the attachment of this flagella to the bacterial cell, you can see there will be three parts. The first part is known as filament, extended from the cell surface to the tip. So it is the external part that we called it as a filament. Basal body, it is the embedded in the cell. So the bacterial cell, where you can see the basal body is there, and that a basal body is embedded in the cell wall and cell membrane. To these two parts, that is basal body and the filament, there is a one connecting part that is known as hook. So it is a hook like structure, and that's why the name hook we called. This hook is having the further attachment as filament, and basal body is also attached to the hook. So hook is the middle part of the filament or flagella. The basal body of gram-negative bacteria has four rings connected to the central rod. So when we observe this central rod, you can see that there will be four rings that are connected to the basal body. A pair of ring embedded in cell membrane, M ring and S ring, and another pair of associated with cell wall, that is L ring and P ring. So these are the gram-negative bacteria having the four rings, two rings in a cell membrane and two rings present in a cell wall. The outer L and P rings associated with the lipopolysaccharides and peptidoglycan respectively, and the inner M ring plasma membrane and S rings in a plasma periplasmic space is also made up of the protein that we called it as a peptidoglycan structures. Now here this is the diagram which is taken from the book. Now here you can see hook structure is given over here. So the hook is the topmost portion which attaches the two different parts, the filament and the basal body. So L ring you can see here, which is present in outer membrane. Then P ring is there, it is present in peptidoglycan region. S ring is present in a plasma membrane, and M ring is also present in a plasma membrane. So these are the four parameters which is present in the bacteria, and this particular structure you can see here. In a gram positive bacteria, hook is present. Then the in a peptidoglycan region you can see there will be a presence of hook like structure. It don't have any ring like structure, so two rings are present, and that two rings are present in a plasma membrane. So this is nothing but the simple structure. Now gram positive bacteria, what possesses in a gram positive bacteria? They have only two rings in their basal body, the inner M ring connected to the plasma membrane and the outer S ring attached to the peptidoglycan layer. So here we can say that this is nothing but the discussion about the locomotory organ which is present in the bacteria. So watch this video 2 times to understand the concept of locomotion in bacteria.